no demoniaco, bueno.
Ah,
No demonio.
no, sí sé, pero. No, sí se lo va a ver.
No. Ya estuvo conversando. Magtox. Magtox.
Ada Max. Ay.
No estaba el proyecto. Sí.
Ah, entonces
Por ahora no me ha
Fair.
Regresando a la India. De la India estaba. De la India. ¿Ah? ¿Qué pasa? Ah, no, no hay que ver.
Que no sé cuál es la sigla 7. No te conocí.
Yo tengo mis caballos.
Reinaldo, ¿cómo lo hacemos para que salga por YouTube? ¿Cómo lo hacemos para que salga por YouTube?
¿A dónde lo busqué?
O sea, sí, pues la
Lo que pasa es que tiene que estar de acá.
Aquí a marcar tarjeta. Su marido está muy popular. Sí, así que vamos.
Let's not. Just do it. It's not.
Qué bien.
Hasta
poco me decía, no, desapareció el programa de acción en la academia.
Y sí,
No sé cómo por qué, ¿no? Y tú le vas a comprar. Sí, no quiero ser específica.
siempre hay como 1%
Estamos en el 33% los recortes. Sí. Obviamente, ahí van a recortar.
¿Se nos permite?
Eso es muy social. Pero igual, que andar un poco la. Pero no la sirven totalmente, porque ya existen las que son.
Ya, hola, buenas tardes. Estoy muy contenta porque veo hartas caras acá. Significa que el tema es importante para varias personas acá en la facultad, además del magíster. Para las personas que por primera vez vienen a estos seminarios, estos son una parte importante de las actividades presenciales del magíster en toxicología. Y siempre tenemos este tipo de invitados tan importantes que cubren algunos temas que nos vemos durante las sesiones de clase, pero en realidad muchos de estos temas después, si despiertan interés, los vamos incorporando también a nuestro quehacer. Voy a presentar entonces con mucho agrado al doctor Muhammad Abbasi. Él es farmacéutico clínico de profesión y su especialidad es farmacólogo clínico y farmacometrista. Tiene entrenamiento avanzado durante su doctorado y en etapa postdoctoral. Y su especialidad es la modelación PK-PD y PBPK para optimizar las terapias farmacológicas, especialmente en poblaciones vulnerables, como neonatos y pacientes críticamente enfermos. Tiene bastantes publicaciones de alto impacto y al mismo tiempo participa activamente en enseñanza y ha trabajado mucho con estudiantes de farmacia también. Su trabajo se centra en la dosificación de precisión y en la farmacoterapia basada en la evidencia para mejorar la seguridad de la medicación y los resultados en los pacientes. Así que les pido por favor que demos una bienvenida al profesor y the floor is yours.
Thank you very much. Good afternoon to everyone. Today's topic is regarding this, regarding the PBPK, which is physiological based pharmacokinetic modeling. So, I believe like most of them, you know the pharmacokinetics. Pharmacokinetics is very important in to know the drug concentration in our human body or in animal bodies. So, toxicology is mostly important. Why? Because it producing the side effects in the higher potential that causing a very severe effects, maybe it could be fatal. It's not only just a simple side effects, because simple side effects like suppose someone take the drug and they're having the rashes, we can manage it, but someone is having the kidney damage with the drugs on the first dose, it is too much fatal. So toxicity is very important. Today we are going to discuss about the definition or the background and the introduction of the toxicology. Then we'll move and check out how we can use PBPK modeling, and we can find out the drug concentration in different organs, because most of the time we just we focus on the drug concentration or the chemical concentration in our blood, but we don't know what is happening in some other tissues like kidney tissues, and constantly exposure of the chemicals and drugs can get accumulation in the organs, and unknowingly it is damaging our organs. So now here there are two things of exposure. One is like you're taking drugs and for a long time, like suppose a patient is taking the blood pressure lowering drug. We know that this patient is taking this drug. But what about if we are inhaling the gases or the chemicals, suppose take example of benzene. So that could be a very, keep constantly depositing in our body and having some problem related to bone marrow. And sometimes keep constant exposing of this benzene could have the cancer, it's called cancerogenic, so it could cause the cancer. So this is mostly happening those people who are working in the industries or they're living in the industry zones. So now this toxicology is useful for our drug development part, and again in the exposure of the drug via environment. So we know the toxicology is the study of the poisons, and when there is exposure of chemical, we could have the two kind of toxicity. One is the local toxicity, which is on organ level or tissue level, and another you have the systematic, which is causing whole your body. So that could be harmful for any part of our body. These are the two important things. So now you know what kind of the toxics are there. There could be drugs, there could be chemicals, there could be chemicals in the air, there could be some kind of radiations. So these are the substances or the exposure could have the toxicities. So what are the challenges in toxicity? Because previously we have to do the animal experiments, and we had to sacrifice a lot of like animals. So now the regulatory agencies and the things are changing, and now we are most focusing on the silico methods. We are using next generation risk assessment methods using our computational power and our mathematical understanding used to call system biology, because system biology scientists, they are working how we can capture our physiology in a mathematical equation, or we can find out some trends. So we can have the idea without doing a real experiment. We could find out doing our computational power. So in that, I just would like to say, previously we have to do the toxicology studies using the pharmacokinetics, simple pharmacokinetics, one compartment pharmacokinetics. But now there is a PBPK and QSP. I'm not talking about QSP, that is a quantitative system pharmacology. But today the scope of presentation is physiological based pharmacokinetic modeling. But I'd like to differentiate. In QSP, we are not focusing the physicochemical part or physicochemical part of drug. In QSP, just we focus on the mechanistic way. However, in our PBPK, we contribute the physiological part, mathematical part, and biochemical part, and we unite and interlink them to find out the concentration of or the chemical exposure in our body. So this is something I would like to say. Previously, we are doing heavily dependent on the in vivo studies, but the things are changing. Now the solution is the PBPK modeling. So with the help of PBPK modeling, suppose if you want to know the concentration of some tissues in brain, or what is the concentration of drug in the brain. Most of the patient is having some, they inhale some gases, and that could have an impact on their brain cells. And we don't know exactly what could be the concentration in the brain. But I would like to say one thing, that PBPK modeling or pharmacokinetic modeling or pharmacometric modeling is data-based, data-driven. So someone has done the studies previously and they publish in the literature. So we have to do a more data mining, because if suppose if one is working on a PBPK modeling, he's supposed to do a literature review a lot before he have to build the model. So now coming back to evaluation of toxicology. So now we are getting this PBPK modeling method to what you call to determine the concentration or the concentration of drugs or chemicals. What is the PBPK modeling? PBPK modeling is nothing but we are using a mathematical equation and we are linking the physiology of the patient and the physicochemical properties of the drug and what is our objective. Suppose if we have given, if we want to know the concentration of drug in a human, suppose we haven't did this before doing the studies. Like suppose drug is in the part of drug development and it is a preclinical stage. So what we want to know what could be the drug concentration or what could be the plasma concentration of this new drug. So we have the in vitro data. What is the in vitro data we have? We have the physicochemical properties of these drugs. What is the pH, what is the partition coefficient, molecular weight, and physicochemical properties. With the help of that, we can predict the drug concentration in plasma. So with the help of PBPK, we can do that. Now, another thing is why we have to do this PBPK modeling. Means previously we are doing the simple PK, but simple PK is not that much efficient or useful to solve the critical and very complex question. So that's why PBPK is very important. Suppose we have the data in the rat or animals, but we want to know what would be the first human dose. So with the help of PBPK using in vitro, in vivo correlation or extrapolation, we can find out what could be the dose or what could be the drugs. You might know the basic parameters of pharmacokinetics. If you know the clearance, if you know the volume of distribution, you can find out the dose. So with the help of this PBPK, we have the concentration, we have the time. Concentration and the time. We use it and we can calculate the clearance and we can calculate the volume of distribution and that we can find out the dose. So this is very helpful nowadays to find out the first in human dosing as well. So these are the applications. We could use this PBPK modeling in drug development. Even we can find out what could be the drug concentration in the pediatric population. If you know the drug development, when the drug is in the first or second stages, we don't go and study in the pediatric population, in the pregnant woman, in the critically ill population before our drug get approved. But if question arises, what could be the dose for the pediatric population? With the help of PBPK, we can find out the dose in pediatric population without doing a real clinical trial or clinical study. So these are the important application of this PBPK modeling. Now I'd like to show how impactful is this PBPK modeling in literature. If you see from 90s till 2026, I just searched a term in our PubMed and I found like every year there is a huge addition of publication in our database. And we are trying to solve a lot of questions with PBPK modeling. That's why you can see our regulatory agencies like USA FDA or environmental protection agencies or EMA from European Medical Agency, they have adopted and they have given the guideline that how we can use PBPK modeling and how we can solve the complex question with the help of this PBPK modeling, and what should be the method. They have given all guidelines and standards to use this PBPK modeling and to submit these reports when they are filing the for drug development. So same thing like USA EPA, when someone is working on the pesticides, fertilizers or chemicals that could be used for human use or the animal use, so they have to get approval from EPA before it's come to the market. So they have to submit the PBPK report of this drug, means how that is exposing to the animals and the humans. So without that, they will not approve their new chemicals. That's why as you see the regulatory agency, they need this analysis reports. Before moving to what exactly is PBPK modeling, I would like to say this is a well famous quote from the statistician George Box. He told that like a very funny thing, all models are wrong, but some are useful. So we cannot capture nature in a mathematical equation. We cannot, but we are trying and and we are trying and making the usefulness of that models. So if you see here, we have a different in vitro studies. So we need that. And from that, we have the in vivo data. And we have different computational softwares. So what we need the complete physiology of human or rat or dog, whatever you want to work on it. And you have the in vitro information, all the physical chemical properties of drug. And now, like I think most of someone has heard that organ chips. Now a lot of agencies are lobbying that don't sacrifice the animals. So what we are doing, we are just doing some tissue assays, and from that we are getting a data, and we are using that data in our softwares, which is having mathematical equation. They are interconnected. So most of mathematical equation we have, that is ordinary differential equations. So maybe in our, I don't know about high school, we have learned about the ordinary differential equation. Maybe 11th class or 12th class, I don't know, but yeah, I have read in my 11th and 12th class. So it is a algebraic equations. So we connect that equation, we write that equation in the softwares, and we connect, and we have to predict the drug concentration or the chemical concentration in tissues or in our blood. So this is the overall sketch of how exactly PBPK modeling can be done. So if the question arises, what exact the component of PBPK modeling? So we need the system data. Whatever the system data, it might be animals or it might be humans. So we need their demographic data, their age, and their physiological parameters, all that kind of information we need. Apart from that, we need to know what is our trial design, means what are our questions. And the drug data. We need to know the log P, what is the solubility, what is the dissolution of that drug, what is the pKa, that kind of information. That is very primary and preliminary information when the chemical is or the drug is in this preclinical stage. We get this data very early. So as soon as we have this data, we can start working on our PBPK modeling, and we can start working in drug development part. When I was working in the industry, so most of the molecules, they were in very early stage, and they start predicting what would be the drug concentration in human. And that is the very useful to design a clinical study, because question arises, what could be the drug concentration if it is oral dosing, if the patient having the food, empty stomach, in stomach with food, what is the drug concentration? So that kind of question can be solved by doing the PBPK modeling before exactly doing that in clinical setting. So now we have the model, but can we trust that model? As I said, all models are wrong, so we need to do the validation. So the important part of this PBPK modeling is you have to make the model, predict the concentration, and after maybe 4 or 5 years, 3 years, you did the real study, then you bring that observed data and paste or match that data with the predicted. So if that observed data and the predicted data matches, then we can say the model I made is correct or useful. Now here you can see this is the pure the real model. If you see here, so we have the blood, arterial blood concentration, we have the venous blood, and in between there we have a different organs. So this is the structure where we make our PBPK model. So here you can see all this model we are writing in ordinary differential equation. So you don't need to write all every time because it is by default, but if you have some different mechanism or different output, then you need to change. But by default, you have all the equation in our software. So you need to just input the data and create your questions, and based on that, we can try to find out the answers of our questions. So in PBPK, you will find two kind of PBPK. One is the minimal PBPK, and another one is a full PBPK or whole body PBPK. So if you want to know exposure of the chemical and drug throughout the body, then we can use the whole PBPK model. But some question is like that: if the patient having, take example of a critical ill patient, he is having brain infection, and we have given antibiotic. We are giving antibiotic by IV, okay? And we need some minimum inhibitor concentration of antibiotic in brain. Suppose it may be 4 or 2 mg per liter. So it is very hard. It is very hard to get the concentration from brain in a patient. But at least we need to know what could be the concentration. So what we need to do, we need to bring all the system data which mimic the critically ill population, their physiology. We have to change all the physiology. What is their renal function? What is the cardiac function? All that we have to input. We have all the options that we can put. What is the blood flow? What is the blood? What do you call that? The body temperature? What is their all the parameters we have to give that, and that will give the output. What could be the concentration of that drug in a brain? So if it is suppose it's supposed to be not more than or less than 2 milligram per liter, and what our predicting, then we could say that okay, our drug is meeting the minimum inhibitor concentration. But this is just a model. Until unless we have the real data, we cannot validate. But it will give some idea. So now here, if you see in this slide, you will see all on the middle. You will find all the equations we are using. So these are the equations, ordinary differential equations we are using to calculate the concentration in different organ or different arteries. So this is something like overall structure or the equations we use. Apart from that, I would like to say that PBPK modeling, it is not like one button or like clicking and you are getting all the information. You have to keep repeating it, until and unless you match your objective. Because most of the time when we do the PBPK modeling, we have the system data, drug data, and we have the trial design questions. And same time we have the observed data as well. So we make the model and we match with our observed data. And based on that, we can say that our model is doing well and our model is being validated. So there are two kind of method. One is that bottom up. So PBPK is bottom up, means you don't have anything, but you are predicting the future. Means what could be the human effect, what could be the clinical effect is having in vitro data of chemical or drug. So this is about the way how we do. We have to learn and confirm until unless we validate our model and we can say this model is true and correct and useful. So these are the tools we use to build this model. There are different packages, some are open sources and some are like we have to purchase it. So PKSIM, one of the open source package from Europe, especially Germany. So it is open source, like all the scientists, they come together and they prepare the software for open source and anyone can use it, no need to pay and nothing like that. But some of the like SimSit, which is from Sartara, which is more advanced and more easy to use because we have to pay for that. And we can build many models. Suppose with SimSit, if you want to predict the drug concentration of exposure in the fetus, suppose a mother, a pregnant woman, she has a baby and she's having asthma, okay, and she is taking asthma drugs. So questions are like unnecessary. The drug is getting exposed to the fetus. If it is getting exposed to the fetus while the woman is pregnant, what could be the drug concentration that exposing to the fetus, and is that drug concentration is harmful or not harmful? So this kind of question can be solved with this PBPK model. Apart from that, I just like to give one example of benzene. So this is the most exposed drug, most exposed chemical in the environment, and there is reported having the cancers, and that is affecting the bone marrow. It causes the bone marrow toxicity. So we find the concentration of benzene in the bone marrow, and if this question arises, what concentration of benzene could lead to the cancer, So this kind of question can be solved with the PBPK model. So this was a published study. They work on the benzene and build a PBPK model. Their objective was to build the PBPK model of benzene and try to find out the concentration in human. So this was the model structure. So you can see that our body having some slow perfused organ and some are the rich or highly perfused organ. So what does it mean? It means that if it is slow, means the exposure of the drug to that part is very less or minimal. If it is highly perfused and rich, perfused, the concentration of the drug will be more. Again, it depends upon the drug's physicochemical properties. So here you can see that we have built a model in different organs. We have considered the fat as a one compartment. Sorry, they have considered the fat in one compartment. They have considered livers, and they also has gone for the benzene metabolite. What are the benzene metabolites and what is their exposures in human? So these are the concentration. They made a model and they verified that models with this PCP model. Here, if you see there is a four things you will see. You will see in the solid line, which is the median or the average concentration of benzene and their metabolites, and there are two other dash line which represent a 95% confidence interval. So whenever we are predicting the model, we used to call it is VPC, visual predictive check. So this is the way we validate our model. So you can see that our concentration or the observed data, which is within the predicted concentration, then we could say that the model we built is useful, or we can say is like correct or useful model. But suppose our prediction is and the observed data, they are not matching and not within that 95% confident interval, then we have to work, or we can say the model is not wrong, or the model is not predicting well, or the prediction potential of that model is not true. So this is one of the example that we can calibrate and do the validation. So in PBPK, first thing we have to learn the model, do the calibration, and then we can go for the validation. But even when you are doing the calibration, you have to meet the criteria. The criteria is that the prediction and the observed data is matching as shows in the figure. So it is a visual predictive check. So now this is the validation. If you see the prediction and the observed benzene concentration are within the within or nearby the prediction line. It is not very like far away. However, some of the section, if you see here in a urine, this is a lot figure number C. Then you can see the concentration is little bit predicted concentration is not little bit matching with the observed data. So this will shows that model need to improve more so as we can reach the maximal with the real data. So we need to do the sensitivity test. So what is exact the parameter which is we are using our model is not matching. So sensitivity test will give the optimized value for that parameter. So in the another slide, what the scientists researchers had did, they try to find out the AUC, area under curve, or exposure of the benzenes and their metabolites in the bone marrow, because getting a sample from bone marrow is not that much easy, and we cannot have the concentration. Instead of that, the data was available in literature about the AUC. So when the modeler, they build the model and they mimic the situation or the mimic the case like here that explain that they have given this it says that metabolite of the blood of bone marrow over 24 hours following 8 hours of steady exposure, and they have given the dose, or the dose was like 100 parts per million. So when we are doing these toxicology studies or the environmental based toxicology studies, most of the time the dose is we are not measuring in, usually we do in does in the mg milligram per kg like that. So here there is a 100 parts per million. It is airborne exposure of benzene. So you can see they have tested. They have the data of bone marrow of benzene and the blood. So you can see that the data from bone marrow is not predicting very well when compared to the blood. But this is the best model they can produce based on the data, and they have predicted, and they try to show that if the AUC of the benzene in the bone marrow is little, like up to 100 up to 40 parts per particle ppm, then the model could be best. But if the exposure is increasing, then our prediction is not going to be a correct one, because if you see the figure, then you will understand. So I just want to show there are a lot of advantage, but same time there is a limitation. As I said, that all models are wrong except some are useful. So this is about PBPK. We can use PBPK in clinical settings. We can use PBPK in animal settings, preclinical development, clinical development, and we can find out the concentration. We can find out the doses of that drug, exposures, that kind of things. So this is about one example of benzene PBPK, an exposure to human, and how can we find out the concentration in urine or bone marrow, and something useful, we can do that, and it is very interesting. PBPK PBPK modeling. So this is about PBPK modeling, useful in toxicology. I believe that you have you understand my explanation. Does anyone have any question?
Sí, tenemos unos varios minutos, así que si hay preguntas.
No, primero las alumni.
No, see, maybe it's not about that much of toxicology, but one of my issues sometimes is that when I want to change the route of administration, but I only have the data for like either oral delivery or IV, and what model, or do you think there's actually a useful model to do that change? Because I usually work with inhaled drug delivery. So that is, it's always like, what would be the dose for my inhaled formulation instead of?
Yeah, here if you see the first side is a dosage form. It could be oral, it could be IV, you know. So PBPK will help you to go to back, because our main target to find a dose. You know, when the PBPK has developed, when the pharmaceutical company is failing a lot of drug trial in clinical, because they want to find the right human dosing, first in human dosing, they call for FIH. So translating from animal to human, it is not that easy, because when they are going the first trials, a lot of toxicity kind of reaction is happening, and they are losing a lot of money. So that's why they are working a lot before going to do that. Right. So yes, your question is that regarding the dosage form.
Yeah, I'm changing the route of the administration now.
Yes, that is possible. That is for PVP.
Okay, yeah.
That is for PVP.
Is it a specific type of equation that you would use in that case? Do you know a specific one that is used for inhalation? It might be too specific, really. So.
Yeah, it's
sorry.
But your question is like, if you want to change the route, and what could be the concentration of that drug? Your question is like that.
My dose in as an inhaled instead of, for example, oral delivery.
Yes, we can.
Yeah.
We
Okay.
can find easy ways.
Yes, work together.
We need to work, we can find. Because the most important thing is that if we have a drug based on their physicochemical property, we decide what could be the dose, correct? And what could be the dosage form. And again, suppose if we have a drug and we decide oral dose, but our viability is not meeting the criteria, then we have to change our dosage form. And that's why we have a PVP we can use.
Yeah.
Okay, thank you so much for your talk. I would like to highlight that we don't have anyone working on this area here at the School of Pharmacy. Now my question, which is, do you think it's worthy to include genetical data, pharmacogenetics about transporters or the SIPs, the metabolizers? Or is not clinically relevant
No.
for your
No.
model?
No. SIP or pharmacokinetic data is very important because PBPK deals first ADME, right? Absorption, distribution, metabolism, and excretion. So the pharmacogenetic part is the metabolism part. So if drug is having the metabolite, so there is a role of SIP. And we need to include the role of SIP, means the drug is get metabolized by CYP2926. So what is the contribution of this enzyme? Means how much of metabolite metabolized by this enzyme. So we have to include that. I published my
in the
Yeah.
your equations.
So I just try to show just in general. But we can use that SIPs information.
And also transporters, like PGP,
Transporters,
if you're
PGP, yes.
thinking on the brain, I think it's quite important.
We can include that.
Thank you so much.
Without that, we cannot. For some drug, we need it because, like PBPK works on perfusion and rate limited disposition of these drugs. So there are different methods. Suppose some drug get metabolized a lot. Take example of acetaminophen, which is paracetamol. Then without considering the metabolism, we cannot predict the right concentration of that drug. So there is a role of all these things.
Good.
So any further questions?
What are the principal limitations when you're working with PBPK modeling?
Only the data.
Then,
So either you have to sit yourself and gather the data, read a lot of literature, or someone is bringing data and everything. This is the data, and just use your computational or PBPK skills. And I want the output, then there is no problem. But bringing the data mining is very important. And apart from that, the researcher, they come to PBPK modeler, they ask, they even don't know how to use PBPK, what is the usefulness of the PBPK. So if they are asking a direct and right question, then very easy to work on it. But sometimes if not understanding, then it's a little bit challenging to understand what is the question. But in general, data is the limitation. If you have a data, then you can do. So like here, if I am showing you here, this, I supposed to, like, if I have data, I supposed to, instead of using, it's not me, the researcher, they're supposed to use the concentration in a bone, but they don't have. They have some data publicly about AUC. But the best way is to present the concentration.
So I guess a model is just as good as the data you work with. That's like a general.
Yeah.
Yeah.
There is another quoting in the modeling way, they say garbage in, garbage out.
Right.
So this is that, if you have something nonsense data, the answer will be nonsense and not useful. So because when I was learning these things, my mentors, they told me these two things, that all models are wrong, some are useful, garbage in, garbage out. So
That's
if
right.
you are not focused, then yeah.
Yeah, yeah. So I don't know, are there any other questions, doubts that anyone may have? So, si no hay más preguntas, entonces un tremendo aplauso.
With this, I just, with this, I take the opportunity, if someone is interested, want to do this, not only PBPK, population pharmacokinetics, clinical pharmacokinetics, we can work together.
Voy a pedir a que se muevan, se ve todo muy bonito. Could you sit down somewhere? Because I just want to get a picture. Nunca hemos tenido tanta gente por magíster, entonces. Sonríete para Instagram. Otra más. Muchas gracias. Un momento.
Para que salgas tú.
Ah, pero
Ahora vamos a salir.
Cheese. Cheese.
Una más.
Whisky. Toxicología.
Y la pantalla. Ah, no, está el pino ya, lo sé.
Ya. Muchas gracias. Gracias a todos.
No, yo no tengo.
So like small isolations wouldn't help to like small things to make this nice. Like you have worked like and you know it's neonates, things like that. So
Yeah,
yeah,
building a model is okay, but validation and
Yeah.
and suppose if you are working on a drug, so we need to know physical chemical property.
yeah.
And we can sit one day and I'll explain every.
No, it's
the
okay.
software.
Yeah, it's okay, and you see, it makes sense that you need the data to validate it is what it is, but it's always like, the same way that you try to reduce the amount of benefits. And in this population is so processed, they're so limited, around the world, people don't wanna actually be doing something like clinical trials in. So it's something that you might want to reduce the amount, but obviously in the data,
I'm interested in the question you asked.
Yeah.
Because but they are formulation scientists. So PPK has a role in formulation because optimization of the formulation is the PPK model. So using for biopharmaceuticals and physiological based model.
Yeah, so the thing is, sometimes we, in the lab that we work so much about like drug delivery. And we want to go into animals. We might want also like the quicker answer of like, oh, they want like oral delivery to inhaled delivery, you need to adjust the dose by this or increase it. But it's always like depending
What
on.
is the bio? The
Yeah.
question is the bio.
Yeah, it's a lot of
So one day I was talking with Danya, that formulation scientists are working a lot.
and we do
Pero ahora que conociste el web, ya sabes que estaban armados. O sea, de hecho, creo que sangre venosa, ya infección solo igual lo buscas o tendrías que tener los datos para que las moléculas sí, pero y eso. y las perfusiones para cada uno de los órganos y también hay ahí, pero evidentemente lo que dice la Carolina me dice sabiendo al momento de un dato bueno de algo, pero sabiendo la velocidad de metabolización de ese algo puedes predecir. Y ahí ya después como medicina, una farmacoterapia muy individualizada. Yo podría decir, no sé, por la cinética enzimática de una persona súper distinta a la de otra, y trabajamos con el paciente y ver cómo, o sea, no le decían que hay 500 miligramos a veces. ¿No es cierto? En el fondo, eso quiero decir. Bueno, vamos a los del magíster. Vamos a empezar. Entonces somos chicos, nos encontramos en la puerta por 5 minutos más. Lo que queda de cada encima de las mochilas, como siempre, podemos subir. Oye, esta silla aquí se ve tan fea que estamos sacando fotos de la ciudad. Esta hay que tirarla para acá. 18 horas no te va a pagar, no va a pagar lo mismo. No, pero yo voy a decir al Miguel. Chao, gracias por venir. Ya, entonces los que quieran dejar en mi oficina, puedo dejarla. Yes, you may.
Ya lo que como no sé, pues metabolizado por 3 a 4, entonces ahí para
Entonces, como que yo antes tenía miedo de la mujer. Pero ahora no, ahora me conozco, pero el problema no es que nos vamos a tomar
no, yo creo que súper bien. ¿Sabes cuál es el tema que me pregunta esto? A lo mejor, claro, este financiamiento, a lo mejor como un genérico, porque este de suceso en específico, a mí no me gusta porque había faltado. Yo, te lo digo, yo trabajé en la base.
Sí, no, o sea, sí.
O sea, si no estoy con una mutualidad, antes de si salgué, yo convencí con eso.
Pero que lo que yo refiero es que, como está cocinado, a lo que yo creo que hay que proponerle a la misma mutual la segunda etapa. Eso es lo que yo digo.
Ah, sí.
Yo me voy a seguir con los sucesos, pero seguir con la misma velocidad. Pero en una segunda etapa de un trabajo, ¿cachai?
O se puede hacer otro trabajo con otra exposición, ¿cachai? Eso se puede
muchas como no sabía cómo iba a ser. Y yo honestamente sí, estaba más o menos desde el punto de vista de manejar la plata, de entonces, no, y la gente como bien, o sea, quien nos está preguntando es la gente interesada.
Claro.
Entonces, no hay una auditoría como, ¿sí? Hay son como,
Claro.
en cambio, este sentido que no es así.
Tiene una diferencia.
Entonces, para o sea, por ejemplo, hay alguien que otros proyectos, ¿no?
Sí.
Lo difícil,
No, a partir de la parte de la tarde. Sí, pero igual es que
hay algo que te van acortando.
no te pasa plata para que tú.
Inventes algo,
Claro.
si no le gusta un producto,
Es una idea tuya, ¿cachai? Entonces, a esa idea tuya, él te dijo, ya te la compro, pero después te la cobro. ¿cachai?
¿no? Y demuestra que no está.
A eso me refiero, ¿cachai? En cambio, este otro, no, que este es un problema que a alguien le interesa.
No, y bueno, yo te paso la cara, te doy el tiempo,
Claro.
vamos, pero es distinto.
Sí,
Por eso digo yo, a pesar que,
yo todo.
o sea, que yo no sé, o sea, y la semana del papel, yo no soy la semana del papel, y la semana del papel no me sirve.
Y la ansiedad, ahora me lo explico.
O sea, de verdad, tú no o sea, otros informes que ¿no?
Ya.
Me los devuelven, informes que o sea, es toda la cuestión es como ultra mega detalle y me lo rebotan, y no, porque falta esto,